So, continuing with the methods for immobilization of enzymes, we shall be talking today about the entrapment methods. So far we have discussed various methods under the category of adsorption, including ionic binding and metal salt linkage, cross linking and covalent bond formation. Now, entrapment methods are also a very large class of enzyme immobilization methods that are based on the occlusion of an enzyme within a constraining structure. That means, the enzyme molecules are entrapped within a definite region of space. It could be a lattice of a polymeric matrix or a semi permeable membrane, and now this matrix within which the enzyme is entrapped must be tight enough to prevent the release of the protein and allow the substrate or product molecules to pass through the boundaries. Basically, it means that when we have a enzyme which is entrapped within a gel matrix, the gel should be porous enough so that within the pores it can retain the enzyme molecules which are large molecular weight in size of that of a protein, and but the porous structure must be such that small molecular weight substrate or product molecules are allowed to pass through, and therefore, the reactions can be carried out. Similarly, on the other hand, if the entrapped preparation is like a microcapsule which also consists of a semi permeable membrane boundary, the semi permeability of the permeability of the membrane must be such that the enzyme molecules are not allowed to be leaked out, while the substrate and product molecule can pass through easily. Now, some of the important characteristics of these preparations, as I mentioned just now, that only reactions involving relatively small substrate or product molecules can be carried out successfully using entrapped enzyme preparations, because the enzyme is entrapped, it does not have any modification or involvement or coupling of the amino acid residues or the protein molecule with that of the matrix of the immobilized enzyme preparation, and therefore, the sole force that keeps the enzyme molecule bound to the matrix is the porous network and purely by molecular size, and therefore, only small molecular weight substrate and products can be processed, while the large molecular weight substrate or product cannot be processed using entrapped immobilized enzyme preparations. The major advantage of the process or major advantage of this method is that it does not require any modification or involvement of the amino acid residues of the enzyme, which results in essentially in the high yield and stability, because there is no modification of the enzyme structure involved in the entrapment methods. It is purely a physical sort of restriction constraint of the enzyme molecule within a narrow region of space, and therefore, the enzyme does not undergo any deactivation practically, and usually the yields obtained by the method are usually quite high. Second thing, the enzyme, the microenvironment of the enzyme can also be tailored to suit its stability. I mean, while immobilizing the preparation, one can add substrate molecule or other stabilizing agents, so that they are retained along with the enzyme and stable preparations can be obtained. Two major types of entrapment methods that are usually followed, that have been extensively followed, are by gel entrapment. That means, basically the enzyme molecule is you have a gel matrix, a polymeric gel, and enzyme molecule is essentially entrapped within the lattices of or interstitial spaces of this gel just by physical confinement, and of course, there are a variety of methods to make the gel network. We will discuss that. And on the other hand, the second method is micro encapsulation, where you tend to form a kind of a artificial cell with this boundary of a semi permeable membrane, and the enzyme molecules are entrapped within this membrane, and these microcapsules can be used as immobilized enzyme preparations. Now, looking at the first method, that is the gel entrapment, it involves entrapping the enzyme within the interstitial spaces of cross-linked water soluble polymeric gels. Mind, I think some of the phrases which I mentioned, cross-linked water insoluble polymeric gels. Now, in most cases we start either with a monomer or a water soluble polymer. We start with a monomer unit, and only those cases where the polymerization can be carried out under mild reaction conditions in the presence of enzyme, and the monomer solution, which is usually water soluble, is mixed with the enzyme solution, and conditions are created so that polymerization can take place under mild reaction conditions, of course with the help of a catalyst or initiator or some other conditions, and a network of polymeric gel is formed which is water insoluble, and then the enzyme is entrapped within the interstitial spaces, or alternatively we take a water soluble polymer, large molecular substance, and mix the enzyme solution with this and the mixture is subjected to conditions by which the insolubilization takes place, and therefore it could be either by change of solvent, or could be change of by certain ions, it could be by temperature. There are the usual parameters which can induce insolubility, or it can affect the solubility of the polymer. The polymeric network can be obtained from monomers, oligomers, or polymeric precursors by changing the solubility variables. So, there are two approaches by which the preparations are made: by changing the solubility variables like solvent, temperature, pH and ionic strength, or the cross-linking reaction. Now, if it is a water soluble polymer, one can also induce cross-linking by certain conditions, so that it becomes water insoluble and the enzyme gets trapped into the matrix. One of the most common methods that have been used, in fact, if you look into the whole variety of entrapment reactions.
Sir, when you have solubility in that case, also the cross-linkings are taking place because you are changing the solubility parameters, right?
You see, the purpose is, if we start with a water soluble polymer, it will usually not have any cross-linking. It will be water soluble with lot of hydrophilic groups, and the enzyme can easily mix with the solution. And once you change the once you induce cross-linking into it by any method, it could be by a cross-linking bifunctional reagent like butyraldehyde, it could be ferric chloride. I mean, a variety of methods have been employed, and after cross-linking is induced, the whole network becomes water insoluble, and the enzyme molecules are entrapped in it. On the other hand, when we are starting with a monomer unit which is water soluble, then also to get a water insoluble gel, you also include a cross-linking agent. In both cases, cross-linking, that is why I say these two parameters are important. One is the degree of cross-linking, and other is to change the solubility parameter, and both the matrix, ultimate matrix, must be insoluble in water, and also it should be in a proper range of cross-linking such that the porosity can be regulated. In fact, cross-linking helps you in a very significant manner to control the porosity of the matrix. Higher the cross-linking, smaller is the porosity. And that, now one of the most commonly used method, as I was talking about, is the polyacrylamide gel entrapment. This is polyacrylamide gel entrapment. More than 50% of the total literature cited on entrapment methods would probably be devoted to polyacrylamide gel entrapment, and of course it was one of the earliest methods also, because the polyacrylamide gel preparation was a existing methodology even for gel electrophoresis. So, people use the same method to even prepare an enzyme containing gels. The method is very simple in the sense that we start with an acrylamide monomer which is water soluble. To this, a cross-linking reagent, usually N,N-methylene bis acrylamide, usually often called as bis, I mean it is also called as bis, is added to it, and then enzyme solution is mixed with it. The 3 water soluble material are mixed, to which an initiator potassium persulfate or ammonium persulfate, whatever you can take as initiator, or riboflavin. In fact, a number of initiator molecules can be used. It could be ammonium persulfate, it could be potassium persulfate, or even a riboflavin molecule. They are initiator. That means it is essentially a free radical polymerization. So, you need a initiator molecule which can initiate the free radical reaction, and then TAMET N,N,N,N- matromethyl tetramethyl ethylene diamine is used as a catalyst. And this mixture is now the 2 parameters which are very important to control here is the absence of oxygen. The presence of presence of oxygen inhibits the polymerization reaction. So, it is very important that all the reagents that are being used, they are free of oxygen. Basically, I mean if you just sort of bubble through nitrogen through the reagents, you can get them free of oxygen, but even traces of oxygen can I mean reduce your degree of polymerization in the polymerization reaction. And also the second parameter is that the reaction must be carried out at less than 10 degrees centigrade. Of course, the reaction temperature, while it will control the reaction rate, it will carry out the reaction at a slow rate, and essentially it has also been seen in the polymerization reaction that the slower the rate, degree of polymerization can be taken up to a higher level. And secondly, that it is also important for keeping the enzyme in a good shape, I mean to allow not to allow to inactivate the enzyme, because the enzyme the reaction is exothermic, lot of heat is generated. So, one has to make arrangement for dissipation of heat, so that the reaction can be or the temperature can be maintained at less than 10 degrees centigrade and enzyme is not deactivated. After and it is a very quick, very fast reaction. In fact, the whole reaction is over in a couple of minutes. In a couple of minutes, you get a polymeric network of chains. Now, essentially it is a 3-dimensional network. These are the chains which are shown with the dotted lines, indicate the chain, the cross linkages which are vertical to the plane of the paper. So, it is a 3-dimensional network which is formed, and enzyme molecules are entrapped within this the interstitial spaces which are created by the cross linkages that are maintained, and you get a smooth solid block in the form of a gel. The porosity of the gel can be maintained by concentration of the cross linking agent, that is bis. Temperature also can control the degree of polymerization, and all those parameters can be optimized for given conditions, so that the maximum yield of the enzyme activity is recovered at the end of the reaction.
So, what did you say is the 3-dimensional part, the cross link?
This network what you get is essentially you end up in a 3-dimensional network which is water insoluble and in the form of a block. And what I wanted to state was that you see here some of the cross linkages are shown ending with the dots. Now, these ones are just perpendicular to the plane of paper. That I mean that is what it indicates. So, that just to indicate a 3-dimensional network, and the enzyme being entrapped within the space, and of course, the conditions of the concentration of the acrylamide monomer, the concentration of the cross linking agent, that is bis, the temperature, the reaction temperature are the 3 major parameters which control the degree of polymerization and porosity of the block, porosity of the gel. Now, after the gel is formed, then the gel is sort of mechanically ruptured into small pieces, either in the form of small granules or it cut into small pieces, and then can be used as a packing material in the reactors. In addition to this, another very common methods, common group of method which has been used are the use of photo cross linkable resin prepolymers. Now, these are mostly oligomers which can be polymerized using UV light. A large number of methods are based on the use of photo cross linkable prepolymers. Now, I have shown here some of the reagents like ENT is a water soluble derivative of polyethylene glycol, ENTP is a water insoluble derivative of polypropylene glycol, and ENTB is a water insoluble derivative of polybutadiene butadiene, then PBM is malic polybutadiene, and PU essentially is polyurethane, and these are the precursors of certain photo cross linkable polymers. That means these molecules can be polymerized using UV radiations in the presence of a photoinducer, again a catalyst kind of a situation, and benzoin ethyl ether is used in most cases as a inducer for photo cross linking. And these materials ultimately end up, I mean they can be dissolved along with the enzyme, and then polymerization can be started by exposing them to ultraviolet light at 360 nanometer for a few minutes, and in few minutes the reaction is over, and the gel preparation containing the enzyme molecule is made available. Another group of entrapment methods, gel entrapment method rather, is from the long chain polymers. Now there are two major approaches by which these gel entrapment preparations are made from long chain polymers, preformed polymers. So far as you see, in two earlier methods we have talked about the use of a monomer and an oligomer, a polymer. On the other hand, here we are using a ready made polymer, and the first method, which is also a very commonly used method, is inotropic generation, in which the whole process, the insolubilization of the polymer is based on ionic exchange. Basically the enzyme, water soluble enzyme, is mixed with an aqueous solution of the appropriate polyelectrolyte. Most of the polymers used here are polyelectrolytes, which can be mixed with an enzyme solution, and you get a very typical preparation which has been reported is sodium alginate. It is a polysaccharide with a very large negatively charged polyelectrolyte, and now this polyelectrolyte is then mixed with the enzyme solution, and this mixture is then added dropwise with addition into a solution of counter ions. The sodium is replaced by calcium. Of course, calcium has been most commonly used. So, instead of sodium alginate, you end up in calcium alginate, which is water insoluble. But solely sodium alginate is water soluble polymer, and it can be mixed with enzyme solution, and then this mixture you extrude it through a fine syringe into a calcium chloride solution and agitate it, and by the exchange of ions, the sodium get exchanged with the calcium, you get water insoluble beads. One of the major advantage of this method is that while in the case of previous methods you get the block of the gel, and the gel block has to be mechanically dispersed into a particular shape for use in the reactor. Here you can prepare the beads, the immobilized enzyme preparation almost in the bead form, so that that can be directly used. Of course, one of the disadvantages here is the stability of these material. For example, calcium alginate, although insoluble in aqueous medium, but gets solubilized by certain ions like phosphate ions. If your medium contains phosphate ions, calcium alginate will slowly get sort of dislodged, and you will end up in dissolution of immobilized preparation. And very often, sometime instead of calcium, then barium or aluminum is used as a counter ion, and such methods are very commonly used, not only for enzymes, but as we will tell that even for using some of the other biocatalysts like whole cells. This method provides a very viable method for immobilization. The second method is based on precipitation. That means you change certain conditions in a way that the mixture of the enzyme and the polymer, preformed polymer, changes its solubility behavior and gets precipitated. Now some of the natural synthetic polymers that are used are collagen, gelatin and agarose are very commonly used polymers. Of course, collagen and gelatin are proteins, whereas agarose is a polysaccharide. So, a large variety of natural polymers are used by changing one or more of the parameters in the solution. Now, in the case of collagen, which is essentially a structural protein found in the slaughterhouse waste. Now, this collagen can be taken, mixed with the enzyme solution, and you make a dispersion of the enzyme with the collagen solution. You do not get really a good solution, you get a real dispersion of the enzyme because being a structural protein. And now this dispersion is then casted on a sheet, so that you can get thin film of the enzyme entrapped in the collagen membrane. Now, to provide the stability to this molecule, to this film, usually the film is subjected to cross linking either by glutaraldehyde or by ferric chloride, of which different methods have been used. So, cross linking becomes an important parameter to provide stability to the immobilized preparation. Similarly, in the case of gelatin, one can mix the gelatin solution with the enzyme solution and cool it. Cooling will render the reduce the solubility and we will get a gel, and after the gel is formed, then you can subject it to glutaraldehyde to provide cross linking and therefore get a permanently insoluble gel preparation. The same applies to agarose, which is also a polysaccharide and also can be precipitated or can be converted into a gel by cooling. And of course, there are a number of other polymers, including synthetic polymers, which have been used by this mode into an immobilized preparation.
So, collagen is not suitable.
Collagen is not so good suitable. In fact, that is why I say it forms a dispersion with the enzyme molecule. And now, as a matter of fact, the collagen method is not exactly like a gel as we consider a polyacrylamide gel or an agarose gel or a starch gel. It is essentially a dispersion which is casted into a thin film.
So, dispersion of collagen and enzyme in water.
In water, dispersion of collagen and enzyme in water, which is then spread over a thin film. Usually, the preparations of this are made in the form of thin films, and then of course, to give it stability, they are cross linked. And after it is cross linked, then it becomes a permanent.
Like after you make the film, then you put
right. You just add the cross linking reagent after a film has been made.
So the sodium alginate is a negatively charged?
Yes, alginic acid. Another method, a common, a very useful method again for the immobilizing mostly microbial cells is fiber entrapment. Essentially, the physical entrapment of an enzyme here is achieved by dissolving a fiber forming polymer like cellulose triacetate, which essentially is water insoluble. Now, this is dissolved in a non-polar solvent like chloroform or carbon tetrachloride, methylene chloride or things like that, and this solution, this organic solution of cellulose triacetate is then emulsified with the aqueous enzyme solution. You just stir the two solutions, an organic as well as the aqueous phase, agitate it, and you get a fine emulsion, and this then emulsion is extruded through a spinneret. Just like you make nylon fibers, you pass through a fine spinneret, and these fibers, the extruded fibers or the filaments are then dipped into a liquid coagulant like toluene or petroleum ether. Now, when these fibers are passed through toluene, they get coagulated. The polymer get coagulated with the water droplets, the aqueous droplet of enzyme being entrapped in the fiber. Essentially, you pass the, let us say this is a spinneret. Now, this is put into a trough containing toluene. Now, when it passes through toluene, after residence time in toluene, the polymer, your cellulose triacetate gets coagulated, it is no longer soluble, and then these fibers what are formed, they contain essentially the enzyme micro droplets, which were earlier emulsified into the solvent, into the solution of cellulose triacetate, which are entrapped in the fibers. And now, then these fibers can then be used in a variety of modes in a reactor, like even people have tried to make a fabric out of immobilized cells, which then can be used for a variety of industrial applications. The second group of entrapment methods is on micro encapsulation, and in the case of microencapsulation, as I mentioned earlier, the enzyme is enclosed within a semi-permeable membrane, polymeric membrane permeable to substrate and product. Enzyme does not pass through, but substrate or product must be able to other way. Substrate or product must be able to pass through the membrane in the system. Now, one of the major advantages is that it provides extremely large surface area for the reaction, for the enzyme substrate complex, enzyme substrate contact within a small volume. But the preparations are too fragile, because this semi permeable membrane is very thin, and it provides you a very fragile preparation, and therefore its use in a number of industrial reactors is probably very difficult, because it will not be able to withstand the shear and the mechanical load which a industrial reactor for chemical processing will require. Now, but this method has been very extensively used and very favorably used, particularly for biomedical applications. For example, the reference as I have listed here from by Chang and Associates, which was reported in 1971, had asparagine in nylon microcapsules, and essentially the whole preparation of this nylon microcapsules was in the sense that an aqueous solution of enzyme and a hydrophilic monomer like your NS2 hexamethylene diamine plus enzyme is dissolved in a water in a buffer. Now, this is in a buffered medium that is mixed or emulsified in a water immiscible organic solvent. Usually it is mixed with, for example, in the case of cyclohexane chloroform mixture. Now, this buffered preparation of hexamethylene diamine and enzyme is emulsified in an organic solvent, and to the emulsified hydrophilic or monomer solution dissolved in the same organic solvent. Now, to which you add a hydrophobic monomer dissolved in the same solvent. This aqueous solution is emulsified, and the emulsion is added to a cyclohexane and chloroform mixture, and another hydrophobic monomer, in this case is savacyl chloride, and this is dissolved also in the same solvent, and to this emulsion the hydrophobic monomer is added, and at the interface the two monomers get polymerized, and essentially the reaction gives you a polymer that we know as nylon. So, the chain continues, and you get essentially this is what is nylon, and the enzyme, particularly the enzyme which was essentially mixed with the hydrophilic monomer, is entrapped within the microcapsule, and therefore this method that has been used very significantly for immobilizing an enzyme called asparaginase, which is used as a therapeutic agent for persons suffering from leukemia. One of the major advantages here is that the nylon molecule being non-immunogenic, it provides a much better material for putting into the human system. It does not sort of most of the other like carbohydrates or proteins, they will generate they are known to generate immunogenic reactions in the body, whereas nylon is relatively inert as far as the immunogenicity is concerned, and these capsules have been then used for treatment of leukemia, because most of the patients suffering from leukemia, one of the problem is that they are lack of asparaginase. They lose the asparaginase enzyme, the enzyme which hydrolyzes the asparagine in the system, and this has been used. So, most of these methods, while these methods have a academic significance from the point of view of providing certain characteristic features, but for industrial reactors they are not very suitable. I have found only applications in biomedical applications.
So, how are these microcapsules formed? The reaction is on the interface of the
Right,
droplets.
but what you have done is initially you have taken a hydrophilic monomer,
Yes, sir.
an enzyme solution in a buffer, in an aqueous buffer, mixed it into a organic solvent. Now, so you make a fine droplets, a fine emulsion. Now, to this emulsion we add the hydrophobic monomer dissolved in the same solvent. So, at the interface of the hydrophilic and hydrophobic monomer they polymerize, and the enzyme gets entrapped into, and of course these microcapsules are very fine in size. Essentially the size can be varied I mean from the almost about 2 to 3 microns to 10 microns, almost like a artificial cell, an yeast cell for example, almost they will be the size of the yeast cells. Now, if you look at, we have seen so far a large variety of immobilization methods. Now, if one has to really study various methods, I think one must compare the various methods and to evaluate various methods for a particular job. Then I think these are the parameters which need to be looked into. Now, on the right hand side I have given you the various methods that we have talked about. On the left hand side are the parameters which are important to be compared, and of course there is no method absolutely either reported in the literature which can meet all the requirements. There is no ideal immobilization method, and each method has to be weighed in terms of its applicability. And the parameters that are important: method of preparation, binding force. For example, as I mentioned, adsorption has a weak binding force as compared to covalent binding. If you require a very strong binding force, coupling is a yield, a very important again parameter. Enzyme loading, I mean that per unit carrier how much of enzyme can be loaded, and it's something like your specific activity of an enzyme preparation, soluble enzyme preparation. That means in so much of protein what activity can be carried out. Similarly, enzyme loading in terms of carrier loading. Then the choice of carrier, and with the choice of carrier the regeneration of the carrier is also associated. The cost of the carrier and regeneration capacity are directly linked. Stability of the preparation, and of course another is protection from microbial attack. Most of the entrapment methods have a major advantage of protection from microbial attack, because they are constrained within the four walls of a structure, almost like microbial cells, where all the enzymes or proteins are enclosed within a cell wall and a membrane, and within a living cell the enzyme remains safe from microbial attack. Similar situation can be looked from entrapment methods, and finally of course the overriding factor often comes as cost. So, what I wanted to have a look at that all the methods must be considered in terms of these parameters. Now, before we proceed further, I think another point which should be looked at. Very often we have desired, particularly when we talk of the immobilization for industrial reactors, a major factor is the cost, and in terms of the cost it is desirable that when we produce them on a commercial scale using microbial cells, those enzymes which are intracellular in nature, they could be used almost in the form of whole cells. You need not disrupt the cells and then isolate and purify the enzyme and then immobilize, because isolation and purification is a very cost intensive operation for intracellular enzymes. So, in many cases of intracellular enzymes, efforts have been made to immobilize the whole cell itself, and the immobilization of whole cell is a desirable feature, particularly in the case of intracellular enzyme. In fact, as a matter of fact, a major application of immobilized cells have been in isomerization of glucose to fructose, and the process is only techno-economically viable because it can be used along with the in the form of immobilized cell. If because the enzyme, the glucose isomerase is an intercellular enzyme, and if one has to really isolate, purify and then immobilize, probably the process will become non-viable. Today the process is used industrially, commercially, almost in half of the world, wherever sucrose is not abundantly available as a sweetener for glucose fructose mixture, because the product cost is not very high, and therefore the use of immobilized cells provides a very viable alternative. Now, the conditions that are imposed on the using immobilized cells are that the cell should not contain interfering enzymes catalyzing the side reactions. That means, for example, if you are using glucose as a substrate for a particular reaction, then the cell should not contain the enzymes that can also act on glucose and provide you side effects. Now, of course, glucose is such a substrate, you will all agree that it is very difficult to look at a living cell or a microbial cell which will not have more than one enzyme which will use glucose as substrate. I think the very first reaction itself, for example, hexokinase will itself act on glucose as a substrate and produce the corresponding phosphorylated sugar. So, in that case, the other alternative is that it should be feasible that other interfering enzymes, if there are any, it should be possible to inactivate them by some pretreatment which does not influence the desired enzyme. Now, in the case of glucose isomerase or the immobilized glucose isomerase in the form of whole cell, we take advantage of the thermal stability of the enzyme. The enzyme glucose isomerase is a very thermotolerant enzyme. Of the order that the optimum temperature of the glucose isomerase ranges between 70 to 80 degrees centigrade, and therefore you can take the cells, give a heat treatment, whereby most of the other enzymes which are able to act on glucose are inactivated, but the glucose isomerase still remains active, and then use that heat treated cells for immobilization. And so, either the cell should not contain any interfering enzyme. Of course, there are many preparations like, for example, preparation of fumaric acid from aspartic acid, where of course the enzymes are too specific, and usually there are no other enzymes present in the cell which will be able to utilize those substrate. Then of course, the substrate product should also pass through the cell membrane. Of course, that is a major constraint, because enzyme is located within the cell. So, the cell membrane should be able to transport the substrate and the product across the boundary, otherwise you cannot make the reaction feasible. Now, of course, for immobilized cell, although theoretically almost all methods which we have talked, variety of methods what we have talked, starting from adsorption to entrapment, they have been used sparingly, but the bulk of the methods that have been used, rather used on industrial scale, are based on adsorption or entrapment. And most of the entrapment methods very commonly have been used to entrap the microbial cells, and as you will notice that entrapment within calcium alginate beads, polyacrylamide gel, fibers, cellulose fibers, they have been commonly used methods for immobilizing. The method remains the same as we have talked about the enzyme molecule, rather the entrapment of cells is much more, because of their larger size than the enzyme molecule, their retention in the entrapped methods is far superior compared to the soluble enzyme. Then of course, adsorption methods have been based mostly by ionic binding, because most of the cells they have negative charge on their surface, and therefore any positively charged matrix can be used to adsorb the cells onto the surface, and then but again these adsorb preparations are usually in dynamic equilibrium. That means if it is a growing cell, then part of the cells will attach and some of the cells will be leaking out also. But in the case of a non-viable cells, usually entrapment methods are the method of choice. Now, I think we must here also probably make a distinction between living and non-viable cells. Now, living cells are usually used in those cases for bioconversion processes where either we require a series of enzyme reactions, more than one enzyme reaction is involved, usually with a cofactor regeneration. That means a metabolic pathway is involved with a metabolite to be produced, or else if a single enzyme is desirable, then it is always desirable to use a non-viable cells. So, basically the methods, and for living cells usually adsorption has been the method which has been very commonly used, while for non-viable cells entrapment is the method which has been most widely used, but the technical details of methodology for immobilization remains the same as in the case of enzymes.
So could you just repeat this distinction?
You see, when we talk of immobilization of microbial cells, we could think of the cells which are viable, which can multiply. Now, these multiplying cells or living cells are essentially needed in those cases where the process requires a full metabolic pathway, which essentially means a series of enzyme reactions which are interlinked for cofactor regeneration or for some other metabolite uptake and production. And in those cases living cells are used, and usually the living cells have been immobilized by adsorption method, because when they are living on the adsorption method, although it is a method which regularly leaks out the cells from the surface, but then the new cells which are growing during growth phase, they can also bind to the surface and then maintain a dynamic balance. On the other hand, if a single enzyme catalyzed reaction is the desired feature, then non-viable cells are desirable and they can be immobilized safely in the by one of the entrapment methods that we have talked about. Now, finally, I think having looked into a variety of immobilization methods, I think we should also have a code how to characterize an immobilized enzyme. Let us say by any method, or somebody has reported an immobilized enzyme, an immobilization method for a particular biocatalyst. Now, there has to be a common feature by which the immobilized preparation can be characterized, so that the purpose of characterization is essentially that the method should be reproducible. Anybody, the minimum information should be provided, so that anybody who want to reproduce the method can use it for further work. Now, some of these steps which I have listed here provide these kind of, like for example, when we say general description, I am talking of the reaction scheme, that what is the reaction which you are looking at catalyzing, then what is the source of enzyme and source, which enzyme and from what source has been used, carrier. These three things must be clearly outlined. What is the reaction we are looking at? That means also involves which enzyme we want and what is the source of the enzyme, and the carrier which is required to be used for any method. Then the second step is your method of immobilization. That means how do you prepare, what are the reaction conditions, that means the reaction conditions, and particularly more important is your yield, loss of activity and the activity left in the supernatant. So, all that information must be provided in the methods of preparation. Then the third characteristic feature is physical chemical characterization, which essentially means the biocatalyst shape, mean particle diameter, or wet particle diameter. That means after swelling, what is the swelling behavior of the particle, and the compression behavior. In the columns, abrasion, and of course, if it is to be required to be used in the fluidized bed reactor, the minimum fluidization velocity, and all those properties must be identified for a particular, in fact for a particular immobilized enzyme preparation, and when we report any method. Then the final is kinetics and stability of immobilized biocatalyst. a very important feature which probably dictates the choice of the immobilized enzyme preparation. Of course, as far as the kinetics is concerned, what we are talking about, as we have seen for the soluble enzyme, the substrate concentration versus initial reaction velocity. That means the effect of substrate concentration on initial reaction velocity. That was the major kinetic characterization of a soluble enzyme, which gives you information about Km, Vm and other kinetic parameters whatever are there. And obviously, in the normally by convention we represent the kinetic parameters for an immobilized enzyme or immobilized biocatalyst by K prime m and V prime m. The prime is added just to indicate that we are reporting an immobilized preparation, of course, just as a convention. Now, that information must be known, and of course, as you will notice, we will discuss that further, that the kinetic characterization will imply that the Km and Vm values for an immobilized enzyme preparation might undergo a change during immobilization. Of course, other than the reaction kinetics, there are other physical parameters which get involved, particularly diffusion and partitioning. We will discuss them in detail, and then you diffusional effects, then degree of conversion versus residence time at different initial substrate concentrations, the reactor performance profiles and finally the operational and storage stability. The data on these parameters must be provided to characterize a particular immobilized enzyme preparation. Of course, while the first three parameters we have so far discussed, we will be discussing this in our future discussion on immobilized enzyme on the kinetics. So we stop with this.